Hello everyone, my name is Sebastian Bol. I work as a research scientist at the University of Texas Health Science Center at San Antonio, where I do malaria research. In my spare time, I do cat research at Cowboy Cat Ranch in Mico, Texas, United States of America. Today I will be sharing some of our latest findings of our research on cat-attracting plants. Before moving on, I have to thank some people who helped me tremendously. First and foremost, Gavin and Adrian from the University of Western Australia for synthesizing most of the single compounds and for performing the chemical analyses of so many plant samples. I want to thank the ABS and Emilia in particular for their financial support, which made it possible for me to attend the meeting in person in beautiful Costa Rica. I also want to thank Naoki for all his translations of Japanese articles, and finally my wife Evelyn for her help with the bioassays and for allowing me to use our home as a USDA registered and inspected research facility for several years. When I say cat-attracting plants, most people will think of catnip. The response to catnip appears to be blissful and euphoric, and I personally think it is rather bizarre that so few people actually study this behavior. Now, the problem with catnip is that some cats don't seem to appreciate it. We also experienced this ourselves several years ago with the cats that we live with, and this is what sparked my interest in this field of research. Well, the internet is full of anecdotes about other plants that cats are supposedly attracted to, and silvervine has been mentioned repeatedly in the literature. Bioassays with cats have only been done for catnip. To put things into perspective, what was known about the catnip response when we started with our research about 6 years ago? Very, very little actually. About half a decade ago, the behavior had not been described well. We had no idea why cats respond to these plants in the first place. We don't know why this behavior is only seen in felines. We don't know which molecules can elicit this response, and we don't know which olfactory receptors are actually involved. So since we had cats who did not respond to catnip, we decided to do some experiments. 100 domestic cats, most of them in local no-kill shelters, were offered catnip, but we also offered 3 other plants to them. Each plant was made available to them on 3 days for up to 1 hour, and we were curious to see how many of them would actually respond to these plants. Of the 100 cats, about 70 responded to catnip, which was in high agreement with previous results published about half a century ago, and about 80% responded to silvervine, and half of the group to Tessarian honeysuckle and valerian. Of the 30 cats who did not respond to catnip on any of the 3 days, about 80% responded to at least one of the other plants, and almost 95% of the cats responded to at least one of these plants. So while we had tested 100 cats repeatedly but briefly, we did not really study their behavior. With our follow-up study, where we also were able to test Indian nettle, the emphasis was on comparing the behavioral response to the various plants, but also between cats. So for this study, we tested 6 cats, all living with us, from left to right, top to bottom. These are Harvey, Flinder, Eggeberry, Zappa, Namibia, and Ollie. And for Eggeberry, this will be her 3rd scientific publication. We tested 5 plants, each for 10 hours per day on 10 different days. First, we looked at the response duration. On the y-axis, shows the total response time, and the various plants are shown on the x-axis. Each dot represents data from one of the cats. While no responses were observed for the negative control, which was tea leaves, we did observe robust responses to catnip. Now, interestingly, the response duration to the other 4 plants was equal to or maybe even higher than to catnip. When we looked at the cats individually, we found that some cats seemed to have a preference for some plants. For Harvey on the left, she responded longer to silvervine, and Ollie responded longer to Tessarian honeysuckle than some of the other plants. When we compared response duration to cat-attracting plants between the cats, we found that some cats respond much longer than others. For example, Ollie responded much longer to all these plants than Zappa did. So we see differences in response duration between cats, but what about their behavior? Eggeberry on the left almost always sat down with the olfactory sample and rubbed her chin or her cheeks against it, whereas Ollie almost always near immediately flopped on his side after he approached the sample, head rubbing the sample, holding, biting, and raking it. Zappa on the right showed extremely interesting behavior, as you can clearly see, frequently rippling of her back, head shaking, and also rubbing, which was the most dominant behavior that was observed during the catnip response for all the cats. The rippling of the back was exclusively seen in response to these cat-attracting plants and should not be confused with the disorder feline hyperesthesia syndrome. Even when looking at just 6 cats, many of these differences in behavior between cats were statistically significant. While rippling of the back has been overlooked by researchers in the past, it is important to notice that we see this behavior in 3 of the 6 cats in our study. And we found that the behavior from an individual cat to the various cat-attracting plants was highly similar. So, for example, rippling of the back from Zappa was seen in response to all the 5 plants that we tested. We know that cats enjoy catnip because of the presence of a molecule that is named Nepeta lactone, and this molecule was named after catnip, or Nepeta cateria in Latin, and has not been identified in other plants. So we were wondering which other molecules in the other plants may be able to elicit the catnip response. So we synthesized about a dozen of molecules that were similar in structure or mentioned in literature, and performed bioassays with the same cats who also tested the plants. We dissolved all those single compounds in a solvent called diethyl ether, which we use as a negative control. And those controls were always present when we did the experiments, and after 171 hours of exposure to them, we did not observe any response to the negative control. So first I will show you the results from Nepeta lactone and several molecules that are structurally extremely similar to Nepeta lactone. And as expected, Nepeta lactone on the left was able to elicit the catnip response, and the same was true for AP Nepeta lactone on the right. And all other Nepeta lactone family members were able to elicit the response as well. Now these 6 molecules are all called type 2 lactones, and we found that the type 1 lactones, iridomyrmecin and isoiridomyrmecin, where the carbonyl group is positioned slightly differently, also were able to elicit the catnip response. Now it becomes even more interesting. Dihydroactidiniolide, or DHA, as shown in the graph, is the only single compound we tested that, as far as we know, is not exclusively synthesized by plants or insects. Dihydroactidiniolide has also been detected in glandular excretions and the urine of the red fox. And we see that this molecule has the same effect. The behavior that we observed in response to dihydroactidiniolide was very similar to all other cat-attracting molecules and plants. Finally, we looked at actinidin, and this is not a lactone but a pyridine because it lacks the oxygen atom and instead has a nitrogen atom, which makes it structurally the most distinct molecule. And when we analyzed the results for actinidin, we noticed a rather deviated distribution among the cats. And when you look at which cats responded how long to these compounds, you can see that the 3 cats who did not respond to actinidin typically respond quite long to all or most of the other molecules and plants. Now in yellow I highlighted the lactones, and you can see here that cats Namibia, Ollie, and Flinder responded longest to this group of molecules. However, these 3 cats, who are littermates and possibly genetically more closely related than the other cats, showed no response at all to actinidin. We also observed that the behavior in response to actinidin was different. Although head rubbing was still the dominant behavior for Agaberry, she spent significantly more time licking the sample with actinidin. Behavior she did not show for the other single compounds, catnip, or any of the other plants. While our manuscript was in preparation, a group in Japan published a paper, and we found some exciting data in their raw data file. Surprisingly, they did not mention this at all in the article. They looked at the effect of some single compounds on 12 domestic cats and found that while responses were observed to the lactones, 11 of the 12 cats actually did not respond to actinidin. However, there was 1 cat who did respond to actinidin, and perfectly in agreement with our observations, this cat seemed to enjoy this compound more than any of the other single compounds that were tested. So in conclusion, cats seem to thoroughly enjoy many different plants. Those cats who don't enjoy catnip might very well love some of the other plants. Behavior seen in response to these plants is highly variable between cats, but the behavior from an individual cat to the various plants and single compounds is highly similar. Not one, but almost a dozen molecules can elicit the catnip response, and the observed huge differences between cats in response to actinidin may indicate that there are different olfactory receptors that mediate these responses in cats. With that, I want to thank you for your interest in our research. I will spend a few days in the rainforest of Costa Rica hoping to see a wild cat, some birds, maybe a sloth or two. But I will answer any questions as soon as I get back to Texas, and I would very much welcome any comments, suggestions, or ideas for collaboration.